Today, we're going to do things just a little bit different than usual. We're not going to have the entrance questions specific to the way that we do it here, because we have, if you guys have seen or paid any attention at all to the room, you know that we've had a student from UNI up here, and she's been doing a lot of observations, had a chance to get around a little bit. Now she's actually going to do today's lesson for you guys. So she is going to have a specific entrance question going with her information. So instead of me doing it, she's going to have it go with her information so that you guys can still get one, just in a slightly different format. Otherwise, she will be taking over the lesson today, so if you have any questions, please direct them to Ms. Sandman. She will introduce herself to you, give you guys a little background, and then talk to you about what we're going to do. She is now in charge. So.
Alright guys, so I'm Miss Sandman. I'm currently a junior at UNI. I'm studying to be a high school science teacher. And I'm from Sioux City. If you guys know where that is, it's on the other side of the state. And if you have any other questions about me, go ahead and ask. I'm not afraid to answer. But we're going to start out with our lecture, and today we're going over cell organelles. So what I want you guys to do first is please get out a piece of paper, and I want you guys to look at all these cells. Now, I want you to find 2 similarities and 2 differences between them. Now, you can do between all 3 of them, do between 2 of them, and I'll give you guys an example. So, my example is a plant cell has a Golgi apparatus, and a eukaryotic cell also has a Golgi apparatus. And that would be a similarity. So, 2 similarities and 2 differences. I'll give you guys a few minutes to write your stuff down, and then we're going to share and then move on. Yes. Okay.
Hey, Mr. Dick. How's my binder here?
This is my QOD sheet from last time left.
Thank you. I'll take that. You're good. I hadn't seen that with back to the pile of everything over there that I no longer have space for, so put it up there so I make sure we got it back to you. Yeah, no problem.
You ready for my quad?
What?
You ready for my quad?
Right, wherever you want.
You got your hand?
Thank you, actually, I forgot about that. So, we'll just go to the next.
You writing this?
What she said to do the comparison between those similar and different.
Two, pick 2 cells up there and 2 similarities and 2 differences.
So, look at the cells up there. And you can see what I'm even saying. I give the example of a plant cell and a eukaryotic cell. Apparatus. So that would be a similarity. And I wanted you to find 2 similarities and 2 differences.
You don't have to. If you want to, you can. That would be a great way to do it. Organize it for yourself. That'd be a good way to organize it for yourself. That would be a good choice, but you don't have to. You can do it in whatever way you want. Two, but she did sit down with me.
Oh, so that's what
So
that
that's
counts.
like.
he said that the plant and the prokaryotic both have cell walls, so that would be a similarity. Can I have another similarity? Nucleus, so the plant and the eukaryotic cells both have nucleus. Okay, and then what's the difference? Yep. Which one? Okay. The plant cell does have cytoplasm, but so does a eukaryotic cell. I know it might not just say that on there, but we'll learn about that. Can someone else give me another difference? Okay? Yeah. The plant cell has a vacuole. And you can actually say that the plant cell has a vacuole and the prokaryotic cell does not have a vacuole because eukaryotic cells do have vacuoles.
The plant cell has a mitochondria with cytosol, and the other two doesn't.
Okay, so a plant cell has a mitochondria and a prokaryotic cell does not have mitochondria. That is a difference. Okay, so now that you guys have seen these cells and have seen all these big words up here, we're going to learn about what all these are and what they do and in what cells they are in. So first, I just have the types of cells up here, which are the cells that you saw on the previous slide. So we have our plant cells, we have our prokaryotic cells, which are bacteria cells, and we have eukaryotic cells, which are animal cells. So us as humans would have eukaryotic cells because we are technically classified as animals. And I would appreciate if you guys were to write this down because we're going to be doing an activity later that will require you to know. Yeah, go ahead. You can write it down on the same page that you had before. And if you need any more time to write your notes, raise your hand and I'll wait. Need more time? Okay. It seems like most people are done. If at any point you don't get all the notes and your partner has them, just get them at the end of the class. Okay, so first we're going to start off with the cytoplasm and the cytoskeleton. Now another note, I put pictures over on the side because sometimes pictures help people identify what things are. So if you want to, you can draw a picture of it. And another note, with when I say what organelle the cell, like what cell the organelle is found in, feel free to abbreviate. Because I know some people think eukaryotic is a very long word, so just put E or animal for that one. So, the cytoplasm. What it does is it contains the organelles. So basically, it's this jelly substance that holds all the organelles for the cell. And in the cytoplasm is dissolved molecular building blocks, and it has proteins, nucleic acid, minerals, and ions on it. And it's found in plant, eukaryotic, and prokaryotic cells. And then the cytoskeleton is what's used to support and shape the cell. And as you can see from the picture, they're kind of like just tubes. So if you're drawing a picture of it, you can just draw a little bunch of stick lines, because that's basically what they are. And cytoskeleton is only found in eukaryotic cells. the DNA and to have the DNA ready to go whenever the cell needs it. And then the nucleus also holds genes that are used for making proteins. And the nucleus is found in plant and eukaryotic cells. And as you can see in the picture of the nucleus, it has a nuclear envelope which surrounds it. And it also has a nucleolus in the middle. And the nucleolus is the place where proteins are assembled. Next slide. Okay, next we have the endoplasmic reticulum, or abbreviated as ER, and ribosomes. So the function of the ER is it produces proteins and lipids, and as you can see by the picture, it's kind of all just folded membranes that are together. And what else you can tell is that there is a rough ER and a smooth ER. So the rough ER has ribosomes on it, and the smooth ER does not. And leading into ribosomes, the function of the ribosomes is to link amino acids together to form proteins. And the ribosomes themselves are made of proteins in RNA. And ribosomes are found in plant and eukaryotic cells. And if you did want to draw a picture of ribosomes, you could just draw a circle, because that's basically what they look like. People need more time. Yes. And like I said, if you don't get all the information down, you can either ask me or ask your partner at your table if they got all the notes to write it down. But I am going to move on. The next thing we have is the Golgi apparatus. And the function of this is to process, sort, and deliver proteins. And what it can also do is it makes changes to the proteins and then packages them to send them off. And if you look at the picture, it can kind of look like the smooth ER, but what I like to think about is the Golgi apparatus kind of looks like a bunch of pancakes stacked. So that could be a way to think about it. Waffles. Yeah. And then the Golgi apparatus is found in plant and eukaryotic cells. eukaryotic cells. Does anyone need more time? Next, we have the mitochondria. So, what the mitochondria does is it supplies energy to the cell. And because of this, some people call it the powerhouse of the cell. And the mitochondria has two membranes. It has a membrane on the outside and a membrane on the inside. And it's bean-shaped. And then there's something special about mitochondria. Mitochondria have their own ribosomes and DNA. And the mitochondria is found in plant and eukaryotic cells. Next thing we have are vacuoles and lysosomes. So a vacuole is, the function is it stores materials needed by the cell. So it's just like this big storage unit. And what it can store is water, food, inorganic ions, and enzymes. And it's found in plant and eukaryotic cells. Now, with what the structure of them is kind of different in them. So a plant cell, when it has a vacuole, it's like a big overall vacuole. And in eukaryotic cells, it's just like little vacuoles throughout it. And then next we have the lysosomes. And the function of these is they digest and recycle foreign materials. So basically what they do is they get all the bad stuff in the cell and take it out of the cell. So you can kind of relate it to a garbage disposal or a trash can. And they're found in plants and eukaryotes, or eukaryotic cells, same thing. And I put a question mark by the plants because scientists don't really know if lysosomes are in plant cells. And if they are, they're in very limited number. So. I'm going to move on to the next slide. And next we have the centrioles. And what centrioles do is they help in cell division. And that's the main thing I want you guys to know. You guys can just write down they help in cell division. But they also organize microtubules to form cilia and flagella. But like I said, mainly just write down they help in cell division, because that's what's the main thing here. So, you guys know what cell division is. It's basically when you take a cell and split it in half. And in the way that this works is the centrioles go to opposite ends of the cell. They get spindle fibers, which are kind of like tubes, and they connect to the DNA and they split it apart. And then centrioles are only found in eukaryotic cells. We only have one more slide left, so we're getting there. Okay? Alright, last slide. And the last one is cell wall and chloroplast. The cell wall, what this does is it protects, supports, and shapes the cell. And this is found in plant and prokaryotic cells, not eukaryotes. They only have a cell membrane. And then the last thing is chloroplasts. And what chloroplasts do is they carry out photosynthesis. And as we should know, photosynthesis is when the plant gets food and energy from the sun. So it's only found in plant cells. So basically, if the plant does not have these chloroplasts, they will die. So now that we're done with the actual lecture, we're going to do an activity. Now I need you guys to be in groups of 2 or 3. Do you guys want to make your own groups, or do you want me to do it? Okay. Please get in your groups and then sit quietly so I can tell you what to do.
It's like the reverse. It's like personality swap. That's another thing too. Like, don't think, quiet. It's like, what?
Okay. So does everyone have at least 1 or 2 people to work with for this? So, 1 person, or all of you if you want to, please get out another piece of paper, because I don't want you to write this on the back of your notes, because at the end of the period you're going to turn them all into me, so I can see you guys did. So, what I want you guys to do is to apply all of our knowledge that we now know about organelles of cells, and apply them to either an organization, individual objects, or an overall system. Now, I gave an example of organization application because I used to work at Hy-Vee, so that was the easiest for me to pick. Thanks. So, okay, so the nucleus is what holds the information of the cell. So I said that it was the head of the company because as head of the company, you need to know almost everything about it. And then for the ER, it produces lipids and proteins. So what I chose for that was the food court because lipids are fats and proteins are like what we get from eating meat. So like pizza, Chinese food court. And then the Golgi apparatus is what is used to process, sort, and then deliver. So I set the Golgi apparatus to be like aisles online workers, so people send in their orders, we go around the store, find it, then we package it and deliver it to their house. And vesicles is what is used to divide materials. So I said those are shelf stockers. So what they do is they go in the back of the store, take stuff out of boxes, and go put it on the shelf. So it's dividing materials. Then the mitochondria is used to give the cell energy, otherwise it's known as the powerhouse of the cell. And I said that that's the managers because the managers need to go around and make sure everybody's doing their job, kind of gives people energy to keep working. And then the vacuole is what is used to store things in the cell. So I said that was the storage room. And then the lysosomes are used to get all the bad stuff out of the cell, like take it all in and shove it out. So I said the lysosomes are the custodians, so they clean up everything around the store. And then finally, the cell wall is used to support and protect the cell. So I said the cell wall is like a security system. So I just want you guys to do the organelles that I did up here and just give like an object or like another organization or a system to it. Now I don't want you to do high B, and I kind of want you guys to stray away from doing other retail stores. So McDonald's can be different. Just try not to use the exact same things that I use. So if you guys have any questions, just raise your hand. Otherwise, I'll be going around and I can answer any questions.
So, mostly due to the lack of rubber. Dick and I were talking about it working it out. Also, what was it now? So, yeah, no, that's what I think maybe that either they weren't complaining about it or either they weren't seeing it. Lack of time. But is that we'll see how this one works. This is the time where this they could be accomplished or they could not as much because they're a little obviously less skilled, but sometimes you'll know better as you're kind of going around and you're checking up on them, and sometimes that's all it takes is the some pieces, and then they go, not going to get some well,
I noticed that in this class when I was walking around, people weren't doing anything. They're like, oh, but it's not working. It's actually moving around.
for a lot of kids, no, I was reading advanced kids that I
Me or water off.
He said me or exterior. No, but is that going to keep them from working? Me or 90% of the time. And then we're going to try to probably support you need proximity on everybody like that. So you keep moving around, talking to them as much as possible.
Hey, John.
Do we need one paper? We need all do it, or just give me one paper?
You can just do it on one paper because you guys, when you guys are done, you're gonna give them to me.
You know it for right?
Red Foreman? What? Red Foreman? 70s show? Have you ever watched that 70s show?
No.
Oh man.
That was like when I was in high school.
That was like 90s and 2000s.
Right.
Yeah.
Well, that's before high school.
I was trying to remember exactly when, but yeah, no, Red Foreman, his dad, kept calling him. Yeah, they know Foreman, all right. John. You said it's open. Yeah, I was gonna say, but you're good. No, I thought he was.
Yes, I changed.
Yeah. Okay. I was gonna say, what are you doing? Just leaving 10 minutes early.
Happens.
Well, I would say that's it. Right? We're not paying attention, maybe.
Alright. Well, I'm tired of this.
Yeah. I'm ready to be done now. Yeah. I'll go to bed too. Find out first, I'd probably make it for about 4 hours. After 2 days. And I'd be like, yeah, no, I'm okay now. I don't need to do this anymore. I'm done now.
You guys really need help getting started?
Yeah, me too. She got that man. She look like this. She got skinny. You know what she do now?
Like, if you wanted to do,
I just got one more hand, but then she was like, tearing something.
yeah, do whatever you want.
Cut it off. Yeah, I need help for the bug apparatus to make that awesome. Is that a good one? How do you use that? Make that more?
Okay, did you write down what the phobia apparatus can do? Or did anyone?
Yeah.
Different function processes.
It delivers. So, what at McDonald's kind of like delivers stuff?
The food trucks.
Yeah, the food trucks. Yeah, the food trucks can process and deliver. You could also say that like the drive-through workers. Yes. That the drive-through workers can do that. Did you work with him?
I'm gonna do it. I'm like, I think he didn't give me a whole I'm gonna way out. Yeah, he said that when he seen like super and stuff. He said we was hooping into freshmen and stuff. And we was stacked. He said, I said like this, I'm finally happy with him. He said, I was behind him, he didn't know I was behind him. He said, Ramon was doing nothing with me last year. We still losing. Yeah. Coach said we might go up 18 or
Did you see our Wednesday? Hey, Tavius? No. Tavius, is your group on task?
Yeah. Like we almost finished it. The first Wednesday game, when we was down, I think it was Asani and
Your ears over there, on your face. Keep them ears over there.
me, you, Paul, we was in the bench, they was fighting. Asani, Corbin, somebody else. They like actually run us back in the game. We were playing basketball or something. And we came back and they had a shit right.
You can break in the setting.
Ain't nobody worried about Decker. Decker. That's the people. The group's doing this. Join the group.
I never joined them.
You could.
I've been joining them.
Alright, what's up with your efforts today, man?
It's my young puppy.
Yesterday,
I don't know what I did.
All right, can I have everyone's
I can't reach
attention
him
for a
and he
minute?
did like pulled it out of nowhere. He said, "Boom!" I said, "Oh, it's a bit hard."
Everyone look up here, please. So, if you're done, just raise your hand and I'll come around and pick up your paper.
Here you go. We finished with ours.
No, you can keep your notes. You'll need those.
I'm Fraser to be closer, so actually, he organized, but I'm not gonna lie to you. I can tell him I'm not going to first meeting. I'm not going to know. He already said him and Fraser said the first all practice, no dribbling, no shooting. He's talking about dribble or shoot. Like that's gonna be like running. And then we just began to show myself, come on, bro, what can I do to play? And then after that, as Bill started, I was like, oh, we were going to practice, and then we came 10 minutes late. So they said, after minutes late, I said, he's like, I said, you can't go like this.